everyone, today I'm going to be showing you how to do a genetic cross using a Punnet square. First thing I'm going to do is just very briefly go through the ways that genetics will be written for a genetic cross. So for the sake of this, brown hair and green eyes are both going to be the dominant genes. So brown hair is going to have a capital B and green eyes is going to have a capital G. For the recessive traits, they don't get their own letters, they just get the small version of whatever the dominant is. So red hair is going to get a small b and blue eyes is just going to get a small g. Every living organism needs two of each, so two B's and two G's. So it could be two capital B's, two small b's, or one of each, and the exact same with the G's. And this will affect how they look themselves and what they're able to reproduce. I already made a video on genetic terms, so this is just a very brief summary. But homozygous is when two alleles are the same, so two capital B's or two small b's. Heterozygous is when they're different, so one capital B, one small b. Exact same goes for the G's. A dominant trait is the one that you can see the best, the one that comes out, that even if there's only one trait, you can see it. And that's probably the most important part. So if you've got two capital B's, it's going to be brown hair. If you've got one capital B and one small b, it's still going to be brown hair. The recessive trait would be your red hair. So it has to be two of the small b's. There can't be any capital B in there at all, or else it will not be red hair, it'll be brown. So if you're looking at this because you've got exam questions coming or anything along those lines, it's obviously not going to be written out all nice and neat and tidy and easy for you in just the letters. It's going to be written in words. So you might be told you've got a homozygous person for red hair and they're heterozygous for green eyes. So homozygous means that it's two of the same. So if we look back at the sheet that we were looking at a second ago, the red hair was a small b, and homozygous is the same. So homozygous for red hair is two small b's. They're heterozygous for green eyes, so the heterozygous is different. Green eyes was a big G and the blue eyes was a small g, so they've got big G and small g. So then a second individual, because crosses will always give you two individuals, or they'll give you one and tell you that there's two of the same. So this individual is homozygous for brown hair and they have blue eyes. So for this, homozygous for brown hair, again, we know, look at the sheet, it's two capital B's. If they have blue eyes, it means they do not have the dominant gene for the green eyes, so it has to be two small g's. Next, we're just going to cross those two individuals. So we're going to take the genetic makeup, the letters of each one that we got there just a second ago on the other page, and we're going to write them out. What we need next is the individual possible pairs of alleles or genes that are going to make up this individual. So what you do is you can take another pen and you make little sort of, those leapfrog kind of lines you used to do when you were counting on a number line. What we're going to do is the small b can pair with a capital G and the small b can pair with a small g. So we're going to write out each of those. So each pair of alleles should have one b and one g. The exact same for the other one is a capital B and a small g. So with this, you've got your small b and your small g and your small b and your capital G. There's no other combinations you can make with those four letters. The second one is a big G and a big B and a small g, because there's no other combinations you can possibly make with those letters. To do a Punnet square, you need these genes and you're going to write them out and you're going to put a line under it and you're going to put a line to the side. And you're going to put the other genes to the other side. And to get the cross itself, it's very easy. You just have to add them all up. So at the top there, we've got a big B, and to the left we have a small one. So you put both of them down, and then we've got a small g above and a small g to the left. So put both of them down again. Exact same for the bottom. We had a big B at the top, little one to the left, and then a big G to the left and a little g to the top. All of it goes down. So these individuals are going to both have brown hair because they both have a big B. But the top one is going to have blue eyes because it's only got two small g's, whereas the bottom one is going to have green eyes because there's a capital G signifying the dominant green allele. So if these two individuals were to reproduce, they would have a 50-50 chance of either having a kid with brown hair and blue eyes or a kid with brown hair and green eyes. A question I took that I will put credit to in the description because I can't think of where I took it from off the top of my head. It's on my laptop, but it's from a Leaving Certificate higher level biology question, and it's from 2001 7A. So in poultry, feathered legs, so legs with feathers on them, capital F, are dominant over clean legs, so legs with no feathers on them, small f, small f. And white plumage, so white feathers, which is supposed to be capital W, I forgot to write that in, is dominant over black plumage, which would be small w, small w. What will the genotypes and phenotypes of the offspring of each of the following crosses be? So, first thing we have to do when we're doing this is we're going to do part I, so part 1, and we're just going to draw a line across here so I don't get mixed up. We're going to say I. We're going to write down genotype is and phenotype is. So the genotype is what the genetics are. So we can see up here they've already given that to us. Capital F, capital F, capital W, and we're crossing that, that's just what that means, crossing that with a small f, small w, small w. Phenotype is what they look like. So the capital F is going to be feathered legs. So this one has got, I'm just going to write it short, so feathers, and he's going to have white plumage, feathers and white, crossed with, and the two small f's means that it doesn't have the dominant gene, so it's not going to be feathered. So I'm going to write down clean and black, because the two small w's are going to mean that it does not have that dominant gene for white feathers, it's going to have the recessive gene for black feathers. What we're going to do is we're going to look at all the possible things. So from parent number 1, we know that there's the capital F, there's capital F, there's capital W, and there's capital W. So when we cross those together, the only possible genes we can get from that parent is a capital F paired with a capital W. Because we need one of these, and we need one of these. So the only thing we can get is a capital F and a capital W. This parent, again, because it's got two of the same and two of the same, the only thing we can take is a small f and a small w. So when we are crossing these together, we're going to be able to put them into a table like this, where we go capital F, capital W, mixed with small f, small w, draw it up into a Punnett square, and we add them all together. So you've got a big F, big W, small w. So the result is a feathered and white chicken. I'm assuming chicken, I'm going to call them chickens. But it's heterozygous for each thing. So it carries the gene to have clean legs and it carries the gene to have black feathers. Next part of that same question is part II, or part 2. And that is a capital F with a small w, by a small f, capital W. So for this one, exact same thing. We're going to do our genotype. We're going to do our phenotype. We're going to do all the genes that can be in it, which I forgot to label last time. Genotype is exactly what we already have in the question, so we can just copy that down exactly as is. Phenotype is what they look like. So, capital F is a dominant trait. It's got two of them, so it's definitely got feathery legs. So, feathers. And two small w's. Big W is white, so small w has to be the black. So, feathers and black by small f's, so recessive gene. 1 second, the dogs are barking. Had the absolute audacity to be cutting their lawn out front, and the dogs were not happy about it. Sorry. Small f means there's no dominant gene for feathers, so it's going to be clean legs. And because there's two capital W's, it's got the dominant trait, clean and white feathers. So, to do the genes, again, there's two capital F's, so the only possible thing we can have is a capital F. And there's only two small w's, so the only possible gene from him is a big F, small w. Same here, there's only two small f's, and there's two capital W's. So there's no other possible genes for us to take from that. So the Punnett square itself, we're going to have capital F, small w, mixed with a small f, capital W, cross that across and put everything into it. Capital F, capital W, small w. So it's going to be the exact same answer as we got a minute ago, where feathered and white is going to be all of the babies. So feathers and white. Tia, please stop. He's allowed to cut his lawn. Feathers and white, and all the babies are going to be this. So 100% of all babies are going to have feathers and have white feathers at that. Have feathers on their legs that are all white. I'm going to use letters that are easier to distinguish from each other, so I don't like using capital W's and small w's because they look the same when they're written down if you just write them a little bit too big. So I'm going back to my brown hair, red hair, green eyes and blue eyes because B's and G's are much easier to distinguish from each other on paper in handwriting. So we're just going to say, for example, that we've got someone who's homozygous for that and homozygous for that by someone who is the exact same, homozygous for both. So that's our genotype. I'm again going to cheat because I'm lazy. So genotype. Phenotype, that's going to be brown hair and green eyes by the same. So the possible genes we can get from this, there's a big W, or oh my god, I'm tired. Big B by big G, big B with little g, small b by big G, and small b by small g, by exactly the same. So on a Punnet square, that's going to look like this. We're going to have big B, big G, big B, small g, small b, big G, small b, small g, by the same across the top. So to do this, again, you're just adding up the exact same ones. So those are the different combinations that you can possibly get, and they're all going to have just a colour on them to make them much easier. Very last question, I'm just going to shimmy this up a bit. I'm still getting used to the tripod, I keep knocking it down. Part 3 is a little bit more difficult. So big F, big W, small w, by 2 big F's and 2 big W's. So again, exact same, you're going to get sick of me saying it. Genotype, phenotype. I'm getting messy writing because I'm getting nervous and I'm convinced I'm going to make a mistake, so everything is wiggly, I'm sorry. Genotype is the exact same as before. We just copy it as it is. Beauty, stop licking the floor, please, you were just fed. So phenotype of this guy is, he's got a capital F and a small f. So he has the dominant trait for feathers, so he's going to have feathers even though he carries the trait to have no feathers on his legs. Capital W, small w, so he carries the dominant trait, therefore it is going to show. So he's going to have feathers in white, even though he does carry the traits for both clean legs and for black feathers. He visually, his phenotype, is going to be feathers on the legs and white feathers at that. This guy is 2 dominants of that, so he definitely has feathers, and he's definitely white as well because he's got the 2 dominant traits for white. Look, actually a very good example here. Obviously, genetically, these 2 are 2 very different animals. However, visually, they look the exact same. So this is why it's very important if you're ever going to be breeding, especially reptiles and such, that you get their genetics tested, that you sorry, not that you get them tested, that you purchase from a reputable breeder that can supply genetics, and don't just go breeding everything you can, because even though you look at it and go, oh yeah, that's definitely this, it actually might not be. You're like, oh, that's probably just feathers with white and it's got nothing else in it, whereas really it does. This goes a little bit different to the two previous questions. Genes. So this guy, there's a capital F and there's a small f. So what we have to do is we have to join these up so there's always a pair. So what we're going to do is we're going to take this capital F, we're going to pair it to the big W, and then we're going to take the same capital F and pair it to the small w, because that's a possibility that we can have. So big F, big W. Another choice is big F, small w. But exact same, small f is there. So another choice we have is small f, big W, and then small f, small w. So it's basically any possible combination you can have of two letters that are two different letters from this group. All right? And then we're going to be crossing that with a much easier big F, big W. Because there's only big F's there, so we can only take one. And there's only big W's, so we can only take big W's. So then for the cross, what we will do is we will put our FW here, just for the hell of it, because we can make it easier writing out. Can we still see this on the screen? Yes, we can. And we're going to take our big F, big W from here. We're going to take our big F, small w from there. Take our small f, big W. Then we're going to take our small f, small w. We're going to do lines across just so we know what everything is. And then, just like last time, add them all together. So this is a big F and a big F. So big F, big F. Big W, big W. Big W, big W. Same here, big F, big F. Big W, small w. Big F, little f. Big W, and then big F, little f. Big W, little w. So here we have, this is on its own, this is on its own, this is on its own, and this is on its own. They're all completely different genetics. So this one would be feathered and white. This one again is feathered and white. Same thing. I'm losing the will to live. Oh my god, my writing. And then exactly the same. So, yeah, that's gone way down, way down. So, you got a feathered and white chicken with a feathered and white chicken. Surprise, surprise, all the babies are going to be feathered and white. However, this one is homozygous dominant for feathered legs and homozygous dominant for white. This guy is homozygous dominant for feathered legs, but he's heterozygous for the white. This guy's heterozygous, homozygous, heterozygous, heterozygous. So visually, you've got a 100% chance of feathered and white, but none of them are actually all genetically the same. So altogether, you've got a 25% chance of each. Please ignore my writing. I'm writing at such a weird angle. But yes, so that's exactly it. That's how you do a genetic cross. These can get more complicated where, for example, actually I might just do it for the hell of it. For example, there's 4 possible gene combinations here that you need to use, but there was only 1 here. Sometimes you'll have 4 and 4. So you put 4 down the side and you'll have 4 across the top and you'll have 16 different possible combinations of different ones that could be produced from that pairing. I'm after writing that out now with just colours to make it easier. So anything that has a big B and a big G, no matter where it is on it, is going to be brown and green eyes. So brown hair and green eyes. So everything with that I have marked in pink. And in total there are 9 of them. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So brown and green, I have to move this slightly down. So brown hair and green eyes is 9. The next combo we have is brown hair and blue eyes. That's not the best blue, that's a nice blue. So 1, 2, and 3. So brown and blue eyes is 3 individuals. Red hair and green eyes is 1, 2, and 3. So red and green is 3, and then we have one lonely little red and blue. So red and blue is 1. So the answer for this, if it was going to be what's the probability of each one, would be a ratio of 9:3:3:1. And that would be how you do a genetic cross with 2 individuals who are heterozygous for both traits. If you happen to be trying to calculate the possible offspring of 2 reptiles that you have that you're not quite sure how to do a Punnett square to sort that, just go onto morphmarket.com and put in the genes in the appropriate animal, and it'll give you all of the possible offspring and their hets and everything. It's super, super useful. So that's it for this video. Hopefully it helps some people out. If you've any questions, leave them in the comments below and I will do my absolute best to help. And thank you very much for watching.